All right, so I'm very, very happy today to give you sort of the scientific foundation or context in which gene editing exists. What I'd like to talk about today would be first to answer very briefly the question, why do scientists want to edit genes? Second, what is CRISPR? And then to turn to some potential applications and ethical issues, including gene drive and species alteration or species elimination using this technique, species enhancement, and human therapy and enhancement. So why do we want to edit the genome in the first place? The most basic reason we want to edit the genome is to understand gene function. So a basic scientific application. We want to change the sequences of DNA because we have a theory about what those sequences are doing. And then we want to be able to test it directly by altering those sequences. You can also use gene editing or modifications, small and precise modifications of the DNA to eliminate disease-causing mutations. So we know in humans there are a number of conditions that arise from an abnormal or mutant form of a human gene. And if you can restore that gene to its normal state, to its functional state, you could potentially cure these diseases. A third application that's actually quite prevalent in agricultural contexts is to use gene editing to introduce desirable genetic traits into a species. So here, for example, we all are familiar with the fact that if you buy a tomato in the grocery store, it doesn't last in your refrigerator for more than a couple of weeks. And by 45 days, it's in very bad shape and not useful for eating at all. But if you simply eliminate one gene from this tomato, in this case, alpha metasidase, which is an enzyme that participates in the degradation of the tomato's flesh, you can substantially increase the shelf life of produce, which has enormous benefit both for grocery stores and for consumers, as well as for the overall food supply. So what have been the problems with editing genes in the past? In the past, efficient techniques for altering genes only worked in very limited numbers of organisms, particularly well in bacteria. So they were great for scientific applications, but not great for much else. Techniques that did exist that could be applied to other organisms were very time consuming and very expensive, making it difficult to or impossible to alter multiple genes in a single experiment. And most techniques were very imprecise, so they caused a lot of off-target effects. So genes that you weren't anticipating you were altering could become altered as a consequence of your manipulation. So then what enters into the picture is CRISPR, which stands for clustered, regularly interspaced, short palindromic repeats, and CRISPR-associated proteins. So that's very intimidating, but bear with me. When you unpack it, it's actually not that bad. So CRISPR refers to a region of a bacterial genome. So CRISPR is a locus or a region of the DNA where a bunch of short repeats are clustered. So that's the clustered part. The repeats are palindromes. So a palindrome is just a sequence of letters, or in this case, DNA nucleotides, that reads exactly the same in one direction as in the reverse direction. So my favorite palindrome is Madam, I am Adam, which if you read it left to right or right to left, says exactly the same thing. So within the CRISPR region or locus of the bacterial DNA, there are these short palindromic repeats. Those are the little diamonds in this diagram. And they're regularly interspaced by viral sequences, the little ovals that are colored differently. So these are sequences or stretches of DNA that the virus has contributed to this region of the bacteria's DNA. And the Cas proteins are in close, the DNA encoding for those proteins is in close association with this CRISPR locus. So the CRISPR locus contains copies of viral sequences that the bacteria has encountered in the past. And therefore, these sequences are kind of a memory of what the experience of the bacteria has been. And those sequences in combination with the Cas protein is how a bacteria has an immune response to viruses it's encountered previously. And that works like this. So in this diagram, Cas is the little green kidney bean shaped protein. And the guide RNAs, the magenta colored RNA sequence that you're seeing is the sequence that's been contributed by viruses in the past. So it's allows the Cas protein to recognize a region of the bacterial DNA by binding to it. And when these guide RNAs bind to that sequence, the Cas protein then cuts the DNA in half and thereby inactivates it. So Cas will make double-stranded cuts in the DNA based on the guide sequence contributed by the virus, and thereby destroy the virus. So the bacteria isn't going to become sick from a viral infection. The advantage of this system for scientists is that you can use it to make functional mutations in DNA. So you can destroy a piece of DNA's function by cutting it in half. Or even better, you can take advantage of endogenous or naturally occurring repair mechanisms to alter the DNA or edit it by supplying a little pink sequence here that can be plugged into the gap that you've created using Cas. So why is CRISPR such a great thing? Why is it so powerful, and why has it made so much of an impact in the news? Well, the DNA sequence that is inactivated or altered can be precisely controlled by the experimenter, which is an enormous advantage. CRISPR, I didn't discuss this, but CRISPR uses something similar to a two-factor identification approach that we're all familiar with for logging onto our email accounts or other things where we have to answer on our telephone and prove that we are who we are. And having that two-factor system actually greatly increases the accuracy of CRISPR's gene targeting and decreases the likelihood that unanticipated errors are going to occur. So CRISPR relies not on an experimenter fixing the DNA, but on naturally occurring mechanisms of DNA repair to efficiently alter genetic information. It's very flexible. It can be applied to any organism. And most magically, once you have the Cas protein expressed in a system, you can put in as many guide RNAs as you'd like and alter as many genes in a single manipulation as you might want to do, which opens up the door for altering broad interacting networks of genes rather than just one gene at a time in a very laborious and time-consuming way. Moreover, Cas proteins can be modified to have a lot of different functions in addition to cutting DNA. So you can perhaps add a fluorescent tag or a chemical marker to the DNA that you've cut, which greatly enhances the scientific power of this approach. So that's what CRISPR is. How is it that we can use it? I'm going to give you a few examples that can illustrate sort of the power and also the potential damaging features of this new technique that's been developed. So the first thing I'm going to talk about is something called gene drive and how it can be used to alter or perhaps entirely eliminate existing species. So we're all familiar with the fact that mosquitoes carry many diseases. And even though we know that they're a public health threat, repeated attempts to either control mosquito populations or to eliminate them entirely have only been modestly successful. So what if we could actually eliminate mosquitoes altogether? So this has been a fantasy of mine ever since I was 6 years old, growing up in Michigan and tromping through mosquito-infested woods. But it's never proven possible until now. You could, in fact, theoretically do this using gene drive. So if you have a mosquito here shown in red, the genetic mutation, because it has two copies of that gene, the mutation will only be inherited by 50% of the offspring. And what that means is over time, the altered gene does not spread in the population. However, if you use CRISPR, where the cargo in this case is a mutation you're trying to introduce, and you have a guide RNA that will target it to the unmutated DNA strand, very quickly you end up with a homozygous mutation. So a mutation that exists on both strands of the DNA. And in this situation, the inheritance looks very, very different. So a fly that's been engineered or a mosquito that's been engineered to have a double-stranded mutation very quickly passes that mutation throughout the entire population. And in a few short generations, all of the mosquitoes will have the mutation. So how can this be used to eliminate a species? Well, it's actually been tested in the laboratory using a gene called double sex. So this is a gene that in a homozygous state, when there are two copies, uh, the female mosquitoes are infertile, so they don't produce eggs. And what they showed using CRISPR and gene drive in this experiment, whoops, sorry, is that using CRISPR-Cas9 gene drive constructs targeting this infertility gene, it spread rapidly in caged mosquitoes, reaching 100% prevalence within 7 to 11 generations, which is only about 2 or 3 months in mosquito world, while progressively reducing egg production to the point of total population collapse. So this was done in the laboratory, not in wild, but theoretically, you could use the same approach to absolutely eliminate this species from the planet. Which then, of course, raises the first serious ethical concern regarding CRISPR, species alteration or entirely eliminating species from from the world, which raises very serious questions of the impact that this might have on the ecology and the environment. We all know of examples, the introduction of rabbits into Australia, for example, in the 1800s, and the wreaking of environmental havoc that they caused. Um, it's not a thing to do lightly, and yet it's a thing that can be done at this point quite easily. Um, which also raises the question of unintended consequences. We may have unintended consequences on the environment, but we may also have unintended consequences on the species that we're manipulating. And I'll give you just one example. Okay, the example I'm going to give you is a gene called myostatin. So myo is short for muscle and statin means stop. And this is a gene that's been known for a long time to be very important in limiting the production of muscle during embryonic development. So if you have a gene that's normal job is to keep the embryo from forming too much muscle, what happens when you get rid of it? And when you get rid of it, you get what people have affectionately called mighty mice. So if you remove myostatin from a mouse, you end up with these very, very bulky mice that have significantly more muscle than a normal mouse would have, and also a lot less fat. Naturally occurring mutations in myostatin have been observed in both cattle and dogs. So if you're not familiar with the dog on the right, this is a whippet, which normally weighs about 68 pounds, and this dog weighs about 250 pounds. So both cattle and mice and dogs have greatly enhanced muscle production if you knock out myostatin. What's wrong with this picture? It seems like a wonderful thing that we'd want to do for humans if we could do it. And in fact, when the paper originally came out for mice, the authors received thousands of inquiries from the weightlifting community asking if they could have access to myostatin knockout in a jar. And yet it's not as pretty of a picture as it seems. So very detailed studies of cattle that carry this mutation have shown that this enhancement also increases the incidence of many deleterious medical conditions, including dwarfism, syndactyly, arachnomelia, and many multiple metabolic disorders. So we aren't entirely certain why this happens in the case of myostatin knockout, but it is a very negative impact on the individual who was enhanced by this mutation. Which brings us then to human therapy and enhancement. So ethical considerations for human research would include, obviously, safety of the procedure. So we would like to know that CRISPR is very accurate, that actually delivering the compounds that are necessary to do CRISPR-mediated gene modification is a safe procedure, and that we aren't going to encounter unintended consequences like the myostatin knockout. We also need to consider the possible impact of chimerism, or a gene alteration that only impacts some of the cells of the body, but not all of them. And would that impact the ability of cells to work in a coordinated way with each other? A consideration that isn't often discussed in great detail is cost and equity of access. This is going to be a very expensive procedure. And are we going to create two different classes of humans with a significant impact on society, segregating the enhanced individuals from the more marginalized, unedited humans. But the most immediate ethical consideration for CRISPR really focuses on germline modification and embryo experimentation because it's far easier to do these manipulations in early stages of human life in embryos and embryonic cells than it is to do it in a mature human. So, embryo experimentation, species alteration, and commodification of humans are the concerns I'd like to focus on for the rest of my time. So CRISPR was adapted for use in mammalian cells only in 2014, and yet the very next year, CRISPR was used in so-called non-viable triploid embryos, although a significant number of triploid human babies are born alive. So they're not non-viable, they're simply very impaired. Even so, the use of CRISPR so quickly in human embryos prompted a call for a moratorium on further experimentation with human embryos. And yet that moratorium only lasted a mere 2 years. In 2017, CRISPR was used in viable human embryos by this scientist, Shukrat Matalipov. And the very next year, CRISPR was actually used to produce live-born babies that had been gene edited. So in 2018, CRISPR was used on multiple healthy embryos to edit a gene known as CCR5 to a naturally occurring variant that is known to confer some resistance to HIV infection for adults. So HIV is very rampant in China, and the parents of these future babies wanted to give their children an advantage of having this resistance to HIV. From multiple embryos that were manipulated, 2 were born alive, and yet the experiment was done without any animal precedent. So we didn't actually test how safe or healthy this would be before we tried it on human embryos. And there are quite likely to be long-term unintended consequences. And in fact, we know at this point that there are going to be unintended long-term consequences. The very next year, this study appeared looking at the natural occurrence of this gene variant that was intentionally put into these 2 young girls using genotyping and death registry from over 400,000 people in Britain who have this naturally occurring mutation. They estimate that there's a 21% increase in all-cause mortality as a consequence of having this variant. So this manipulation actually created a significant increase in likelihood of death for those 2 girls. Recent studies have drawn into question the accuracy of CRISPR editing, particularly in human embryos. So a review in 2020 in the journal Nature looked at 3 independent studies that show widespread off-target errors in human embryos that had been edited using CRISPR-Cas9. So the first study showed 22% of embryos with off-target errors or unintended mutations introduced using this system. And the 2 other studies showed 50 or 40% errors, including widespread rearrangements of the entire targeted chromosome. So this is very unanticipated. Human embryos, in many respects, have proven to be sort of the exception to the general mammalian rule. We have not seen similar inaccuracies in other species, but we have now seen them in human embryos. So what are my conclusions? CRISPR is a very powerful technique for altering multiple genes in the same organism. Application of gene editing to crop enhancement and pest eradication really does raise global concerns regarding species elimination and environmental impact, as well as potentially unintended genetic consequences. Using CRISPR for human somatic modifications raises concerns regarding safety and equity and social marginalization. And lastly, CRISPR has been used to make germline edited humans with unknown long-term consequences. Oops, and I'll stop there. Thank you so much.
So the topic CRISPR in the law is a massive potential. You could have an entire law school course on this subject. And I have, a small time, so I'm going to limit it. Maureen did a wonderful job of summarizing a very difficult, complex area. I'm going to focus in on one narrow area of the law, and that is, given the way our society is, will people claim there's a constitutional right someday if they can get over these off-targeted problems with CRISPR to eugenically improve your offspring? Or will gene editing take a snip out of justice? That's my one example of a joke. Okay. So why is a lawyer talking to a crowd? I'm not a scientist. I'm not a theologian. And lawyers are talking to, at least in the United States, because of this great insight by Alexis de Tocqueville, who almost 200 years ago, when he came to the United States, the young United States, recognized this reality, which we all live with today. He said, scarcely any political question arises in the United States that is not resolved sooner or later into a judicial question, right? We know that. If it's a political question, if it's a policy question, should we allow CRISPR? What are the parameters? It's going to become a legal question sooner or later. And then combine that with a fundamental case that every law student and lawyer knows, Marbury versus Madison, where the Supreme Court said, it's emphatically the duty of the judicial department, i.e. the courts, to say what the law is. So if a law be in opposition to the Constitution, the court must determine which of these conflicting rules govern the case. And this is the essence of the judicial duty. So again, given our system and given the prominence of the law and the Supreme Court, there is a good chance that someday these questions will be presented. And again, specifically, we could talk about patent issues and all these, but I want to just reduce it to the narrow question, but important question about, do you have a right to eugenically improve your child? So one thing that the law has to deal with, just like the area of science, just like every other area, and that is the area of, or the issue that kind of floats in the culture called the technological imperative. Now, we're all alive because of modern medicine and science. We're not Luddites, we're not against it, but the technological imperative kind of just drifts out there. This idea that if we can do it, we should do it. And of course, there's always a moral question here. The question is not, can we do it alone, but should we do it? As we know from the 20th century, science taught us, physics taught us how to build an atomic bomb, but only morality and ethics can tell you whether you should use it, you should drop it. And it's the same thing with any technology. There are going to be wonderful advancements, it looks like, when you listen to Maureen, with CRISPR, but then there'll be other uses, and you'll have to ask that question. We as a society will have to ask that question, and of course, the law will have to ask that at some point too. All right, here's again the what Maureen showed you, how the Cas9 protein unzips the DNA, makes a SNP, and repairs it, and all the things that Maureen talked about. Now, future of the brave new world and how it impacts the law. So people will start off, if CRISPR can be used more precisely in human beings, to say, well, we're just doing gene therapy, right? We just want to make sure that a deleterious disease or effect, we can stop it from occurring in an individual. And others will say, but we want to enhance our offspring. And then the question will be, is there a fundamental difference between gene therapy and gene enhancement? I mean, will this become semantics? Will this be something that gets, if gene enhancement becomes a germline modification, as Maureen talked about, then it gets passed on to the next generation, and the next generation, and the next generation. We'll talk about that right at the end, but that's a question for the law, because that's a kind of control that you have on future generations. And then the question is, should the law allow that kind of control? Okay, but that's going to be the two basic areas in the law. Probably won't get a lot of opposition if you can say, look, you may have diabetes, but we can do a genetic therapy to prevent you from having type 1 diabetes. You're not going to usually get many people who say that's a bad thing. Again, the question will be about enhancements. But at some point, and the question will be, well, what if the therapy is well, what if we can give you, instead of needing to eat certain kinds of food to get certain nutrients, we can create a genetic modification, for instance, that allows your body to make something that it doesn't need to consume. Now, is that a therapy? Is that an enhancement? So again, I question whether the line between therapy and enhancement will hold, both scientifically and in the law. Okay, let's talk about eugenics, because we're really talking about this. Positive eugenics, of course, is a practice or mindset or mentality intended to enhance a human being. Now, that's a loaded question. Enhance by whose standard, right? What's desirable characteristics? You know, who decides what those are in the culture? Negative eugenics is a practice, mindset, or mentality intended to eliminate a human being deemed physically, mentally, or morally undesirable, or if not to eliminate them, to prevent them from, as we'll see shortly, from procreating. Okay, so again, who decides? What is the standard of perfection that you're using in either situation? The reason that's not just again, a theoretical, philosophical question is because of this case that many of you may have heard about, Buck versus Bell. In the 1920s, eugenics was sweeping the United States, and Buck was a woman named Carrie Buck. She was a young woman. She was born out of wedlock, and she, at a young age, had a child out of wedlock. Bell was her treating physician. And while Carrie Buck was being treated, he was suing to have her forcibly sterilized, because in the mindset of eugenics sweeping the country in the 1920s, certain types of people were the wrong type of people to have children. And if you were promiscuous at the time, you shouldn't continue to have children. Now, this case went all the way up to the United States Supreme Court. It was decided by one of the most famous Supreme Court justices in the United States history, Oliver Wendell Holmes Jr. Now, someone once said, before you can commit homicide, you have to commit verbicide, meaning before you can do something that kind of contravenes the natural law or human nature, you have to kind of justify it in your own mind. So Holmes is going to make 5 moral judgments about Carrie Buck, and the reason he will uphold forcibly sterilizing her, but he has to get there. So this is the analysis. He says, we have seen more than once that the public welfare may call upon the best citizens for their lives. It would be strange if it cannot call upon those who already sap the strength, first moral judgment, of the state for these lesser sacrifices, often not felt to be such by those concerned. Why? In order to prevent our being swamped with incompetence, second moral judgment. So Carrie Buck saps the strength of society, and her kind we get too many Carries, the whole society will be swamped. And then he goes on to say, it's better for all the world if, instead of waiting to execute degenerate offspring, third moral judgment, or let them starve for their imbecility, fourth moral judgment, society can prevent those who are manifestly unfit. Now, it's 5 moral judgments in 2 paragraphs. That's hard to do. And then something that strikes a very familiar refrain right now, the principle that sustains compulsory vaccination, he says, is broad enough to cover cutting the fallopian tubes. And then the money quote, the shocking quote, really, he says 3 generations of imbeciles are enough. Carrie Buck's mother, Carrie Buck's daughter. Now, this is a shocking case. It should shock you, and has never formally been overruled. I don't think the Supreme Court would ever enforce it, but it was cited in the early 2000s by a Circuit Court of Appeals in a string citation for the proposition that sometimes the government can force you to be sterilized against your will. Again, I don't think that's what the Supreme Court would hold, but it sits there. Okay, now, what are some legal issues related to CRISPR? Well, let's start from a state wanting to regulate certain aspects of CRISPR, let's say more eugenic applications. Well, what would be the basis of a state regulation? Well, I would say that just generic police power of a state. All sovereign entities have the power to pass health, safety, and welfare statutes. And the fact that embryos, if you were going to do research on an embryo or CRISPR on an embryo, the fact that today, women have a constitutional right, which of course may be reversed by the Supreme Court in the Dobbs case in the next 2 months. But even when you have a right to abortion currently under existing jurisprudence, if you want your child, states can charge 2 homicides if a drunk driver or an assailant harms a pregnant woman. So the fact that under Roe, you're not a, quote, a constitutional person for purposes of the 14th Amendment and abortion has nothing to do with the fact that states are free and states do protect human beings from the moment of sperm-egg fusion all the way to natural death. So I think that would be the basis for a state regulation of CRISPR. Now, is there a federal or state constitutional right to use CRISPR, whether it's for disease mitigation, therapy, or for eugenics? And I think the argument you'd have to make, and we'll talk about it, is either the contraception cases or the abortion decisions since 1973. One other area would be, is there a right to receive info? Would a physician have to give informed consent about, oh, your child may have this problem and we can take care of it by using this new technology? Again, the third part is the same as the beginning. Do the contraception and abortion cases have any impact on this emerging area of the law? I would say it's extremely doubtful, as we'll see in a second. How about issues of consent? Can parents consent on behalf of their embryonic child? And then issues of equality and justice. These are some issues that Maureen hinted at also. So privacy language, if you look at the contraception case, first one back in 1965, Griswold versus Connecticut, you had the court saying, well, there's no enumerated right to use contraception, but there's an unenumerated right that flows from the penumbras and emanations of the Bill of Rights or the 14th Amendment. Somewhere in the Constitution, this has been pretty severely criticized, and I don't think the court would do that. This Supreme Court would do it, but it sits out there. In 1972, when the question was, could unmarried couples use contraception, the court did throw this language again. I think I question whether this would have any impact on the right to eugenically improve your child. But the court said, if the right of privacy means anything, it's the right of the individual, married or single, to be free from unwarranted governmental intrusion into matters so fundamentally affecting a person as the decision whether to bear or beget a child. Now, again, bearing or begetting a child is one thing. Bearing or begetting a eugenically improved child is something completely different. Again, when the Roe court looked at trying to figure out where the right of privacy is, they threw everything up on the wall. I don't think that the current Supreme Court would go through this substantive due process analysis and come up with a right to use CRISPR, but who knows? It's not clear. Now, I want to talk about the Nuremberg Code that came out of the Nuremberg trials after World War II and the evil Nazi experimentation that went on. There were 10 ethical research principles that were derived, and the first one, is very clear. The voluntary consent of the human subject, i.e. the human embryo, is absolutely essential. This means that the person involved should have legal capacity to give consent and should have sufficient knowledge and comprehension of the elements of the subject matter involved as to enable him to make an understanding and enlightened decision. Well, obviously no embryo can do that. And the question is, can a parent do it? And then you'd have to get into the whole area of what's the best interest of the child. And given the uncertainties and the unknown and the unintended consequences that Maureen talked about, this seems to be a problem. Now, the Nuremberg Code is not necessarily enshrined in state statute or in federal law, but it certainly sits out there as an important guide. And again, another interesting point too is if you were modifying through CRISPR gametes, who's the patient? I'm not talking about an embryo here. So there's no analog in medicine or law for a potential patient or a potential client, not in the way that CRISPR would create it. So just an interesting issue. Now, I'm going to end with two slides. Besides the normal rights we think of with respect to CRISPR, this kind of power over our genetic future reminded me of a passage that was cited by Leon Kass in his book, Life, Liberty, and the Defense of Dignity, where he cites Hans Jonas. He said, and again, forgive me for reading, but it's a very powerful point. He says that there can be, and mostly is, too little knowledge has always been realized. But we always understand our lack of knowledge is usually a problem. But he goes on to say that there can be too much of it stands suddenly before us in a blinding light. The ethical command here, entering the enlarged stage of our powers, as we have this incredible technology, is never to violate the right to that ignorance, which is a condition for the possibility of authentic action. Or to put another way, to respect the right of each human life to find its own way and be a surprise to itself. And I think if either through CRISPR or any technology, if you could find out the day of your death, that's how clear we got to. Would you want to know it? Would you want to know it? And would the law actually protect you from knowing too much? Just again, interesting thing. And then I'll end with this. This is a long quote again, but it's very powerful. This is from C.S. Lewis. This is quoted again by Abolition of Man, quoted by Leon Cass. Lewis says this, and I'll end here. In reality, if any one age really attains, by eugenics or scientific education, the power to make its descendants what it pleases, all men who live after it are the patients of that power. They're weaker, not stronger. For though we may have put wonderful machines in their hands, we have preordained how they are to use them. The real picture is that of one dominant age, which resists all previous ages most successfully and dominates all subsequent ages most irresistibly, and thus is the real master of the human species. Man's conquest of nature, if the dreams of some scientific planners are realized, means the rule of a few hundreds of men over billions upon billions of men. There neither is nor can be any simple increase of power on man's side. Each new power won by man is a power over man as well. Each advance leaves him weaker as well as stronger. In every victory, besides being the general who triumphs, he is also the prisoner who follows the triumphal car. Meaning, do we have a right not to be dominated, right? And can society act to make sure that one living generation doesn't have this kind of control as if you were a commodity only? Again, lots of things for thought. The law is nowhere ready for this reality, but we're going to have to learn how to do it. All right, thank you very much.
Maureen and Nick have already started talking about the ethics of all of this. And so I'm really going to kind of step back for a moment and stop talking about CRISPR per se and try to think a little bit about where we are as a culture. It turns out that science and technology are the products of a culture. It turns out that the law is a product of culture. And so I want us to kind of think about technology in general as a product of culture and how it begins to shape what we imagine reality is like. So as with any novel technology, CRISPR begins with some interesting questions, scientific questions, but the scientific and technical aspects or the legal aspects, to me, they're important, but they're probably not the most fundamental questions, because really what is at stake is the question, what are humans? When we get these fundamental technologies that can transform us, as many technologies can do, we start to ask these questions. So what is it we imagine ourselves to be? What would we have to believe about ourselves in order to believe that CRISPR technology is legitimate? So the problem with all novel technologies is to cut through much of what we think about ourselves. We need to stop thinking of technology as tools. In fact, I would say that this little thing right here, this is not a technology, it's a product of technology, because technology is a way of thinking. It is a logic of technique, or a logic of techne, or a logic of artifice. And so it's a way of thinking. And so these technological ways of thinking already have an assumption about what a human is, but then once they are deployed, they always change the way we think about ourselves. And so I'll just give you a kind of cursory example that's non-scientific, and it's sort of obvious. Before the rise of and the ubiquity of the printing press, the human animal was really much more of a musical animal, because we had to communicate large swaths of information, not stored in our writing techniques, right? But rather stored in our memories. And it's so much easier to remember things if they are musical, or if they have a meter to them, right? And so The rhyming scheme or the meter scheme of a great epic poems permitted people to memorize swaths and swaths of voluminous material, like the Iliad or the Odyssey, or even parts of scripture. So after the printing press, everything starts to change. Of course, this is gradual. It's not an overnight sort of thing. You don't have to memorize everything anymore, right? And you now need to have good eyesight in order to be able to read things, right? And you have to have a very long attention span to read these large swaths of text that really you don't have the sound or the meter that's kind of running through your head the way you do if it's performed orally. So what we see then is that we have a kind of technology, writing and then the printing press, that then begins to change the way we think about ourselves, begins to change what we think, in fact, right? So you no longer have to have a good memory. But with artificial intelligence and novel technologies, you may not even have to have a very long attention span. In fact, some people would say that our attention spans are shrinking by virtue of the technology. So the technology is a product of culture. It is culture that begins to shape us, and to not only to shape us in the way we think about ourselves, but to shape what we think about ourselves, okay? So the most interesting thing about CRISPR to me is what is this question? What do we have to believe about ourselves in order for this technology to develop? And how will it change the way we think about ourselves? So it goes something like this, right? CRISPR technology, I would say, has us thinking about ourselves in a very mechanistic sort of way. It imagines that we are merely material beings, that our genes are really just information riding on the materiality of nucleic acids. It implies in the popular imagination, if not in the scientific imagination, at least in the popular imagination, that one gene controls one trait. Everybody knows that's not true, but it kind of imagines that, right? Myostatin controls one trait, muscle, right? No, it turns out, as Maureen pointed out, it has ripple effects that we haven't even imagined, right? One gene controls one trait. It's kind of a trope in our culture. So we know that's not true, right? So even though we know the idea that one gene controls one trait is not true, there is still a kind of material mechanistic reductivism that is built into first the technology, and then the way that we deploy that technology. And then there's a result from that. The result of this simple-minded way of thinking feeds into a cultural attitude that technology can solve a problem, right? First, we imagine ourselves as isolated agents intervening upon a very small, limited thing, such as a small little tiny gene that can be easily manipulated. But if you know what the gene does, so this thinking goes, and then you know that I can control that gene in some way, then I imagine that I can fix the problem to get that gene to function better, to work better, and then all would be well, or so the story goes, right, in the cultural imagination. But this kind of technological optimism begins to escape, or as Nick called it, technological imperative. The optimism becomes an imperative. And this technological optimism escapes any one particular technology and becomes part of a kind of the whole of technological optimism, right? The way we think and culture today. And it believes that all our problems are merely technical problems, little tweaks that can be made along the way, right? And in time, we think we'll overcome those little technical problems. And we see this sort of attitude in people like Elon Musk. And there's even the attitude that if technology does create a problem, then we will create new technology to solve the problems created by technology. So success in one technology suggests success is inevitable in all sorts of domains of our lives. Thus, we come to think about a gene as an isolated thing, and that our intention can be deployed upon it. And coming out on the other end of that is a success without there being any other effects. Intention is directed at one thing, and only that one thing result kind of enters into the popular imagination. Thus, we become very optimistic that we can achieve that one thing we imagine, and we are optimistic that only that one thing we imagine will result. And then we imagine that we could have laws that will somehow control those one things. So those one things happen and all the bad things don't happen. So this double kind of optimism that we will be successful at the things we want to happen and that all of the imagined or not yet imagined consequences won't fall from it, right? We go ahead and we think, hey, let's give it a shot. And there's even an attitude that says, hey, if it fails, we still learn from our failures, right? You hear that in venture capitalism, which is intimately tied up with technology all the time. Try things, fail, learn, fix the problem. So the result is kind of a cultural attitude that with failure we learn, and that means that we begin to imagine the things we are experimenting upon become to be seen as expendable, right? Oh, well, failure is part of this. So we have these failures. Well, those things are expendable, right? In other words, we don't even have to imagine what it's like for the mouse that has all of those extra muscles to walk around and to live, or the dog, or that bull that we saw there, right? We don't have to think about those kinds of consequences because it's expendable. In other words, we begin to believe things about ourselves that if we are not perfect, then what is the point of living, right? If I can't have those extra muscles or whatever the imagined thing that I need is to make me a better human being, whatever that better means, right? We start to think, well, if I can't have that, then it might be worth the try because this form of existence just ain't worth it, right? And so we begin to devalue the body or to think of it merely as expendable matter upon which we can do whatever we want as long as we're achieving what our intended consequence is. And if we fail, somebody else will learn from it and we will make the species better or something like that. And thus human limitation becomes intolerable to us, right? So that even limitations are imagined as something to be overcome. My terrible eyesight, my nick in my gray hair, right? We need to do something about this, right? And thus we have a technological culture that wants to cross beyond human limitation. And I would say then to cross over or to transgress the human itself in order to enact some vision about some good life, some godlike being that sometimes is referred to as the post-human or the post-human God. So here now becomes the most important question for us to entertain. We're talking about, this is supposed to be about CRISPR and human dignity. And I want to turn to this one concept for a moment and ask the question, given the enormity of the power of the cultural imagination, can a concept such as human dignity push back against this highly cultural attitude that we find with technology? Dignity is, after all, a rather spiritual concept, right? We're dealing with material stuff that we can manipulate, right? But dignity is a rather spiritual concept that says there is something that transcends the materiality of the human being, that elevates the materiality of the human being. Yet it is also a spiritual concept that says that somehow the integrity of that being is so sacrosanct that certain limits ought not to be crossed over or transcended or transgressed. But again, the problem is, to a culture that imagines the best is yet to come, that all of these frailties are problems that need to be overcome, the human itself becomes to be seen as a failure. And I'm not sure that this concept, this spiritual concept of human dignity, can do the kind of cultural work that we want it to do. And even in the law, I don't know that the law can do that kind of cultural work with the concept of human dignity, which, as I've said, is more of a spiritual concept. How does the law make that material? Nick may have some ideas, but I'm not sure it'll work. And then just last November, Alistair McIntyre made the case that human dignity is not a concept that can do the work at all in this culture. And his reasoning was essentially that human dignity as a concept is too reductive and too individualistic. And it therefore has multiple different kinds of definitions, okay? Instead, McIntyre turns to the concept of justice. He suggested that dignity is too individualistic, at least as it's practiced in our culture, and therefore has a kind of corrupted status in our culture. So if our dignity is somehow dependent upon or rooted in our frailties, and it's a spiritual concept, then that means that as an individual, I am probably dependent upon you in so many different ways, including an agreement that there is something spiritual that seems to be the most important part about being human, okay? And thus this reductivism of individualism then is not sufficient. And McIntyre suggested that dignity doesn't do the work, and that the better concept is probably justice or the common good, because the common good suggests some sort of interdependency, right? That is to say that even before my ability to act as an individual, y'all are there to give me culture, to give me language, to give me spiritual concepts like dignity or justice, right? But then I don't know that justice as and the common good are concepts that do any sort of work as well in this culture. And this is where I find us to be in a bit of a conundrum. And so I wonder if we need to start. The one thing I think that justice and the common good does that McIntyre points us to is to show this interrelatedness and this interdependency that we have on one another. And I think that probably needs to be broadened out beyond just the common good as understood as in a sort of social justice sort of way. In fact, I am not only interdependent upon you, right? I am interdependent upon a lot of other beings in this world. Even my own biological existence participates in a different kind of being that requires me to have food, that requires me to have water, that requires me to have all these other things. And I wonder if we need another cultural shift to begin moving away from the individualism that we imagine that technology kind of reinforces to a richer concept that's more ecological in nature, that moves us towards a culture of participation. In other words, we all participate in one another's being biologically, socially, culturally, psychologically. And then that itself becomes the concept par excellence, an idea of participating in divine being as well. That is going to have to make its way into the culture. It's going to require a cultural shift. And I personally don't know how to make cultural shifts like that very easily. And so I'm going to end here, and we can possibly talk about how this idea of participation might play itself out in our culture.
Great. Thank you, Maureen and Jeff and Nick. This was a really stimulating conversation, and we have a lot of questions. I want to lead off with Jeff, your invitation to talk about participation and a cultural shift. So given sort of where we are and where the science is and where the law is, how should we start thinking about what ought we to do both now and in the long term to facilitate such a shift? That's open to everyone.
Well, I'll let Maureen answer this, because I actually think science is one of the places that we could actually go, because Maureen, as a scientist, knows that we're not talking about some sort of linear linkages between gene to trait, right? It's always a gene operating in a biological milieu, and then a gene operating in a cultural milieu or a social milieu that results in something like hypercholesterolemia, for instance, right? So it's not merely this, oh, I've got the bad gene, right? It's a gene that's not functioning in a certain way, or is not functioning at the rate that someone else from another culture or another genetic heritage has a gene that operates. They can eat all the red meat they want. Maybe I can't, right? So it's always genes operating in a biological and a cultural milieu that result in this. And that means that if we tweak one of these genes thinking, oh, we're just going to get this one little thing happen. And oh, mastery, technology, we've won the day. As Maureen pointed out, there are these ripple effects we couldn't even have imagined, right? There are more ripple effects than there are the intended effect. And that's something that science has to say and say out loud and say often, I think.
I think one of the problems with science saying that often and loud is that it's often not true. I mean, there are plenty of genes that have single effects. We know what beta hemoglobin does. We know it binds oxygen. We know it does that in a very straightforward way. We know what happens when we knock it out. We know what happens when we put it back in, and no ripples. So because there are so many examples of that type, I think it's harder for people to appreciate developmental genes like myostatin, that because they impact a unfolding process that involves a lot of interactions, then those pleiotropic or ripple effects can come about as a consequence of the fact that this is a gene that's utilized during this complicated process. But adult housekeeping physiology genes are what we know the most about, and that has set the precedent for one gene, one function. So I think it's to some extent a matter of education, but I believe that the education has to go much further than the question of genes. I think honestly, the ecological example you raised or alluded to is a much more apt model for thinking about it, because there's a tremendous amount of evidence that genetic variation, for example, is incredibly important for the health of the species, right? That we don't want everybody to be, pick your favorite basketball or football player or whatever, to be just that way, because then you lose all the other advantages of all the other phenotypes that exist and all the other traits that exist. And certainly from a Christian perspective, I can remember as a young child encountering somebody who had a very significant speech impediment and being very distressed by this and asking my parents, well, why does this person not speak clearly? And I remember my mother just looking at me and shaking her head and laughing and going, well, that person doesn't speak clearly to give you an opportunity to learn how to be patient. And, that actually applies in a lot of situations. It isn't just that person has a trial and we want to try to fix it. What happens if we never have an opportunity to learn how to be patient with each other? What if our expectation becomes everybody has to be perfect? And I think, as Nick said, who defines perfection? Is it the tyranny of the now over all future generations?
Yeah, I didn't mean to overstate the case. I mean, yeah, of course there are single genes that have single functions, but the repercussions of the loss of those single functions have ripple effects beyond. In other words, we could approach any multiple problems from that biological one gene, one function, replace it, or we can fix the problem or attenuate the problem culturally too. So an example might be, yeah, we can do operations on people's backs because their spinal cord is not functional, and that's what they did with spina bifida and things like that. Or we can actually just make it a little bit more convenient for people to move around in wheelchairs, and a lot of the disability rights community has been saying, look, you're trying to make this a biological problem about my body, when in fact it might be a problem that if everybody kind of found a way to absorb some of this, there are ripple effects culturally that we could do that would attenuate the living poorly in that way. So I didn't mean to say that all genes are not, I just meant to say the ripple effects reach way beyond merely what the gene, what is functioning and what is not functioning.
I agree.
Thank you. So if we could return to a component of all three of your presentations, when we talked about this technology and its possible therapeutic, function. We've had this question asked 3 times. What distinguishes a therapy that might be morally illicit in this kind of context from a modification that would be unethical? One of our 3 questioners talked about editing and you'll excuse me, CFTR and cystic fibrosis as a possible platform for discussion. But first and foremost, Maureen, we have a question. Where do these, when scientists do these experiments, where do these embryos come from? So if we could have some clarification with where the embryos come from first, and then this line between therapy versus an illicit modification.
The vast majority of embryos, perhaps all that are used for scientific experimentation, are embryos that were produced for assisted reproductive technologies and are subsequently donated by the parents for scientific research. It is, of course, theoretically possible for people to use donated eggs and donated sperm to make embryos in the laboratory precisely for experimentation. And somewhat surprisingly to me, there's still a great deal of hesitancy to do that. To my knowledge, it's not being done. So then the question of the difference between therapies and illicit therapies and modifications. You know, everybody's got their own take on this. And I think my own personal take is that a therapy is a restoration of natural function. So if you have a gene modification, a naturally occurring mutation that impairs or restricts the healthy function of a gene, restoring that DNA sequence to a naturally occurring sequence that would have natural function is therapy. Modifications, and I think would be illicit so long as the issues of safety and access and stigma are adequately addressed. The challenge is that between that, the clear cases of restoration of natural function, and enhancement or illicit modifications is an awful lot of gray. There are, for example, modifications, naturally occurring modifications or mutations, variations in gene sequence that give those individuals very significant metabolic advantages, right? They do better for energy metabolism or for cholesterol management or other things. So those superior forms of a natural gene would conferring those forms onto everybody in the population be a therapy? Or would it be a mandate? Whereby at some point you weren't allowed to have poor functioning metabolism. You had to have the kind of metabolism that's the best possible metabolism we can have. And what does that do to natural genetic variation that we know is so valuable in the overall health of the species? And then, of course, the question of unintended consequences. You know, the gene variant that was put into the twins by the Chinese experimenter actually has effects he didn't anticipate. So I don't have a pat answer. I think therapies clearly things that fall into the domain of restoration of natural function would be therapeutic. Other modifications are in that continuous gray zone and at some point cross over into actual species alteration. You know, if we can confer upon people the components of photosynthesis so they can stand out in the sun and make their own energy like a tree does, you know, that could be pretty cool, but it would obviously be a very radical change in the nature of humans.
And the problem is that there's no operating concept of human, a natural human function, right? There's because we again, part of what I didn't say, and then this idea that technology makes us think that we're just a series of information bits that we can then kind of add information in. Well, what's to say that through technology, the natural dimension of human is not standing out in the sun, being able to generate our own energy? We wouldn't have to consume as many, I don't know, beef or something like that. We could actually have kind of constantly renewable source in order to, as long as it doesn't affect natural human function. Well, what is natural human function now? Is it photosynthesis, or is it the kind of way that we think, or that which we name the spirit, or what? So, I mean, I'm not sure that while I, as a physician, when I practiced medicine, I was always thinking, okay, now does this going to return natural function or not? And I liked that because then I had a kind of a rule of thumb. But I started finding that, well, we created a new disease because we found a side effect from a drug that was used to treat pulmonary hypertension in neonates. That new drug is Viagra. Suddenly Viagra comes on the scene, and suddenly what was a normal part of aging in men was the kind of loss of the possibility of attaining and maintaining an erection. Suddenly we have a drug that side effect was causing these preemie babies to have erections, and somebody said, hey, let's use this for impotence, which had been a kind of a loss of natural function. Well, now we've got this pill, we create a disease called, erectile dysfunction. And now we've got because we had a therapy for it now. Right? So what, I'm like, oh, well, is it natural functioning for an old guy to not be able to get erection? I don't know, loss of natural function. Or is it, what becomes natural here, right? So I always wanted Maureen's answer to be right. I just don't know that we have a robust concept of human nature that could put the brakes on other kinds of things that we might find problematic, other than just erections and photosynthesis. That's the part that becomes difficult.
This whole discussion of CRISPR and the science, the profound societal issues, and the law remind me a little bit of three other related areas of technology. One was IVF. And it's interesting that IVF and surrogacy are almost completely unregulated by law. And I, doing pro-life law, I would say that was probably the pro-life community in 1978 when Louise Brown was born from IVF. The whole focus of the pro-life movement was a constitutional amendment. And so just not focusing on IVF, even if you thought whatever a person thought about it, they just didn't focus on it. So that was one thing. Then this area also reminds me of the debate about 15 years ago that's kind of died down about the difference between embryonic stem cell research and adult stem cell research, and human cloning, and could you clone an embryo to produce, to get the embryonic stem cells that you wanted, or would you implant someday? And again, while a few states have acted in the law to regulate or to ban certain technologies, most states haven't. And so it seems to me that we're at a crossroads. CRISPR is just beginning. We're kind of like at 1970, 78 with the issue of IVF. And again, it's pretty much the Wild West in the sense that you can do pretty much whatever you want. And it's very hard to convince people, for all kinds of reasons, to regulate something that you can say that the process violates the natural law, but if you see this baby you want, these are hard issues. Well, I think if serious discussion and thought doesn't go into the issue of CRISPR now, we're going to miss the opportunity. Now, I'm not saying I have an easy answer. Someone asked me today, I was discussing this talk tonight, and I said, they said, well, could you draft some legislation, and how would you draft it? And drafting legislation is always an interesting exercise because you have to get as precise as you can. And that raises all the questions that Maureen and Jeff, raised. I mean, if you use the word therapy, how would you define it? When does therapy cross over into enhancement? How would you define that? Americans, for better or for worse, are very pragmatic. You can ban some kind of research, but all of a sudden, if people are getting X, Y, or Z of what they want out of it, it's going to be repealed, or there's a strong concern about that. So anyway, I just listening to this conversation, it just was kind of a little deja vu from and I think since I'm speaking to the McGrath Institute, is that my got that right. Hopefully I get that right. You know, at Notre Dame, there's a book by law professor Carter Snead that came out that basically makes the point, and I could have gone that way too on this. You know, what's your, this is both Maureen hinted at it and Jeff, what's your philosophical anthropology, right? How do you conceive of the human person that the law is regulating, or that, you know, CRISPR is acting upon? Are we just biochemical machines? If we are, then we're going to treat you differently than if we're, you know, if we believe that, as Western society did hundreds of years ago, that we're made in the image and likeness of God, we have an eternal destiny. We're not just biochemical machines. We might be those, but we're much more. And I think the law, just the understanding of the human person is so deficient in American constitutional jurisprudence that we're going to have to take what Professor Snead said seriously about discussing it in the law or in statutes. So I don't, and you know, this is a fascinating, I love listening to Maureen and Jeff. I could listen to you, both of you, all night. I would be against any of these modifications, but if we could create something that where you automatically made scotch in your body, I might support those. And we could just sit there and drink scotch and talk about these issues because they're fascinating. I mean, they go right to the heart of society. And obviously, very few people even realize that this is what the potential is. And again, we don't want to be Luddites. We don't want to be against science. But how do we thoughtfully approach this? And so I applaud the McGrath Institute for having this discussion, because this is exactly what needs to be done. And again, I'm against cloning, but I might say if we could clone this presentation or presentation like it in 100 different places, that would be great, because this is the kind of discussion that needs to happen. Intelligent, rational, not screaming, no emotion, just talking. What does it mean? What's the implication of all these things? So I applaud all of you for creating this, and I'm very grateful for the invitation.
Thank you, Nick. And we're going to give you the last word. We will be cloning this presentation. It will be available within the week online in some form, whether audio or video. And please share it widely. I want to thank our panelists for an incredibly rich conversation. I think we could have probably just one whole Conversations That Matter series on CRISPR and modifying the human person. I want to thank Christina and Aaron, who have been working behind the scenes to make tonight run smoothly. Thank my co-host Heather, and thank all of you for participating. Whether this is your first one or your 9th one, we're grateful to have you join us. You will be receiving an evaluation form after we wrap tonight, and we appreciate your feedback, and we'll be in touch with you about next year's CTM series. So thank you all so much.
Thank you.